let's just jump right into reviewing. I know I haven't named this yet. But okay. Okay, so gated ion channels, axons, neurons, closed versus open due to receptors, synaptic transmission, voltage gated, ligand gated. I don't really need to know any of this. I already took the exam. I'm really behind on doing these reviews for myself. I'll just do the study guide, wherever that is. Okay, diffusion through the lipid bilayer depends on the hydrophobicity of the molecule. So molecules that are more hydrophobic go through the plasma membrane easier. And water can as well if it's in a small enough quantity. And that prevents passage of polar molecules, osmolality, isotonic, same osmolality outside as inside, osmosis, diffusion of water, yep. Loss of body fluids leads to dehydration, yeah. If you're going to give like a IV infusion, you want to do the same level as whatever the body is at, or if they are out of, say they're in acute shock, you're going to put more sodium because it sequesters fluid from interstitial compartments into the intravascular space. Interesting. If they're too salted up, like if they've got too much salt, then you'd put just water in. Versus if someone's just dehydrated, you want to infuse them with the correct concentration that's equal to their body concentration. Okay, aquaporins form specialized hydrophilic transmembrane channels for the passage of water. Exocrine glands produce sweat, saliva, tears, and those all secrete water through aquaporins. Simple diffusion, passive, yeah, doesn't use ATP. Facilitated passive diffusion uses a carrier protein. That's like when you're transporting glucose across the membrane. Olivia Batten. There's your shout out. Okay, transmembrane protein. Yeah. Differences between ion channels. GLUTs are the specialized receptors that mediate glucose transport down the concentration gradient. You're welcome. And that's in type 1 diabetes mellitus. The inability to release insulin results in low rates of glucose uptake into muscle tissue. And that leads to high blood sugar after a carbohydrate-rich meal. Chloride, bicarbonate, antiport, active transport, sodium potassium pump. Yeah. 3 sodium out, 2 potassium in. 2 types of active transport. Primary, secondary, glucose, symport. Okay. There we go. Voltage gated, channels closed, open, inactivated. Yeah, okay, blah, blah, blah. We're done. Next. Dang it. Gotta move this. Okay. Intermediate filament microtubules are the building block of tubulin. Nope, sorry. Tubulin is the building block of microtubules. Polymers formed from heterodimers of the alpha and beta tubulin. Okay, provides structural support, provides the basis for several types of subcellular motility. Alright, grows more rapidly at its plus end. When it switches back and forth between growth, hey everybody, Cannon and Trish, hi. That's called dynamic instability. And GTP hydrolysis, dynamic instability, rate of tubulin addition relative to the rate of GTP hydrolysis. Okay. Dynein and kinesin provide the force necessary to move membrane-bound organelles through the cytoplasm along microtubular tracts. Kinesin-bound vesicles move from microtubular minus ends, originating at the centrosome. Anterograde fast axonal transport. Retrograde transport. Thin filament, microfilament. It's a polymer of actin. Treadmilling, thick filament. Lysosomes. Lysosomes are like the cell's trash digester. Peroxisomes contain oxidases, and I think they oxidize fats. Yeah. And they also detoxify poisonous substances like alcohol in the liver. Mitochondria could create ATP. Endoplasmic reticulum, the rough ER, makes new proteins. The smooth endoplasmic reticulum makes steroid hormones and other hydrophobic molecules, drugs, toxic chemicals, yeah. Golgi apparatus, like packages proteins for transport, I think. Modifies proteins. Sorts and distributes them to the lysosomes, secretory vesicles, or plasma membrane. Clathrin and calveoli are good for endocytosis and stuff like that. Nucleus makes RNA. Chromatin is a complex of DNA and DNA binding proteins. Yeah. Okay, we're done with that. Now, immunology. Nope, that's not what I wanted to do. There we go. Okay, non-encapsulated lymphoid tissue. It's also known as malt cells. I forget what it stands for. Whatever. A phagocyte will express innate detection receptors, collectively called pattern recognition receptors, or PRRs. T cells do not recognize native proteins. They need an MHC to present them the protein. After activation, a B cell will produce antibodies which are secreted forms of a BCR with the same antigenic specificity. A T cell will express numerous copies of their unique TCR on the surface or the membrane. And that's multiple copies, not just one. Multiple, remember that. They've got their cell is surrounded by those. MHC molecules present antigens to T cells. Okay, cytokines and chemokines, it's basically a communication system. They're like immunological hormones that act in an autocrine, paracrine, or endocrine manner. They orchestrate immune responses. They go where they're needed and perform their function as required. Yeah. A cytokine is a soluble messenger protein released by one cell to bind to a specific cytokine receptor expressed by the same cell. That's called autocrine. Or a nearby cell, which is called paracrine, or a distant cell, which is called endocrine. It usually goes through the blood in the endocrine version. And that results in functional changes to the target cell because it activates intracellular signaling pathways. Okay, cytokines. Cells only express one cytokine receptor, and they have many copies of those receptors, not just one. Cytokines are potent molecules. A cell may produce multiple cytokines, and multiple cytokines may act on the target cell. A cytokine may have multiple actions, that's called pleiotropy, and that depends on the nature of the target cell and immunological circumstances. Multiple cytokines may have similar actions, that's called redundancy, because if you've got multiple, then one might be redundant. You only need the other one. But it helps because if one gets destroyed or something, you can have backup. Cytokines may also act in conjunction, having a concerted effect, and cytokines may antagonize and cross-regulate others. Gene duplication happened as a result of these cells adapting over time. And that's what led to the redundancy. Again, if a gene's missing, that organism may still be fine because there's all the replacements in the body. Cytokines, they regulate, they orchestrate. Hematopoiesis, which is the creation of blood, development, activation, and suppression of immune cells, and direction of the immune responses. Some examples are interleukins like IL-1, IL-2, IL-5, IL-13. Colony stimulating factors like GM-CSF and GCSF. I'm never going to remember that. Interferons like interferon alpha, interferon beta, interferon gamma, interferon tau. Yeah. Tumor necrosis factor, TNF alpha, and chemokines. Chemokines are a group of cytokines. Interleukins, interferons, tumor necrosis factor, chemokines. Okay. Innate and interferons. They interfere with viral replication. They are produced by most nucleated cells. They induce antiviral proteins, and they inhibit viral replication. They increase MHC1 expression. Their main role is to serve as a warning system to induce antiviral states in neighboring cells and enhance immune defenses. Type 2 interferons, which are gamma, are produced mainly by natural killer cells and T cells. They activate macrophages and they boost antigen presentation. And they have a key role in adaptive immune responses against intracellular pathogens. Then there's type 3 interferons, which are lambda interferons. They're similar to type 1, but they're an alarm system. They act mainly on epithelial surfaces like the respiratory system or the gut. And they provide local mucosal antiviral protection. So interferons don't directly kill viruses, but they alter cell states to resist infection. They enhance innate and adaptive immunity, especially against intracellular pathogens. Okay, then there's tumor necrosis factors. These are, they induce necrosis in tumors, as the name suggests. TNF alpha is made by macrophages, T cells, and NK cells, natural killer cells. They're a major pro-inflammatory cytokine. They induce fever and acute phase response. Can trigger apoptosis or necrosis in target cells. Then there's tumor necrosis factor beta, which is also called lymphotoxin alpha. It's produced by activated T and B cells. They promote lymphoid organ development. They support inflammation and immune cell recruitment. And yeah, the functions of tumor necrosis factors are they promote inflammation, they regulate cell death versus survival, and they enhance endothelial activation, which leads to upregulation of adhesion molecules. They help coordinate innate and adaptive immune responses. And these are good to know about because in clinics, too much TNF leads to septic shock, chronic inflammation. This is like rheumatoid arthritis, irritable bowel disease, or inflammatory bowel disease, sorry. And then there's anti-TNF drugs that help if they're out of control. Wow, how much of this do I have left? So these basically are what cause you to feel sick when you are sick. Rheumatoid arthritis, yeah, okay, I talked about that. Some antibodies bind to TNF and block their action, like anti-TNF drugs. And there's currently no veterinary equivalent, though. So animals are kind of out of luck in that regard. Chemokines are a family of small chemoattractant molecules. They recruit leukocytes from the blood and they direct their migration in tissues, guiding immune cells to the site of infection. Chemokines are named according to their structure. Yes. Okay, chemokines are cytokines that control recruitment and migration of cells. Chemokines receive the letter L for ligand. For example, CCl2, CCl3, CCl4, CXCL1. And chemokine receptors receive the letter R, like CCR7, CCR8, CXCR5. And they do not correlate. So that's not helpful. Depending on the arrangement of cysteine residues that form disulfide bonds, this is how they're named. The ligands and receptors do not correlate. Okay, so the migration of dendritic cells from tissues to lymph nodes, the migration of naive B and T cells to lymph nodes, and the maintenance of the organization of secondary lymphoid organs, all of those things as well as recruiting and guiding immune cells to injury sites, those are all done by chemokines. They congregate in sites of inflammation. All right, that's it, I think, for August 27th. Yep. Thanks for watching. Done.